So the last physical method we are going to discuss is filtration. Filtration is a good way to sterilize liquids and gases that are heat sensitive. Rather than destroy microbes, the filter acts as a barrier to retain them. Most available filters only catch bacteria and fungi, but with ultrafine filters, viruses and even large molecules can be filtered. So if you remember from the first lecture, viruses are generally much smaller than bacteria. So you would need an ultrafine filter to catch these. So you have two types of filters, membrane and depth filters. Membrane filters are porous membranes made of organic celluloids, for example, cellulose ester. And although a wide range of pore sizes are available, typically membrane filters that are used to remove bacteria have a pore size of 0.2 micrometers, although smaller pore sizes are available for more specific needs. And the way that membrane filtration works is the membranes are held in special holders, then the liquid is forced through the filter and collected in previously sterilized containers. Then depth filters, on the other hand, consist of a layer of fibrous material that have been bonded into a thick layer filled with narrow twisting channels. The liquid containing microorganisms is sucked through the layer and microbes are removed by adsorption to the surface of the filter material. And by adsorption, we mean adhesion to the surface. So air can also be filtered, for example, by N95 disposable masks used in hospitals and labs, and high efficiency particulate air, otherwise known as HEPA filters, that let air move freely but restrict microorganisms. So HEPA filtered air is essential in a clean room environment, for example, to produce sterile drug products in the pharmaceutical industry. And now we're moving on to the chemical methods of sterilization and disinfection. So we'll go through ethylene oxide, aldehydes, alcohols, phenols, halogens, oxidants, and surfactants. So ethylene oxide is a type of alkylating agent that is used for gaseous sterilization, and it kills both microbes and spores. It kills by reacting with DNA and proteins to block replication and enzyme activity. So it is highly penetrating and can sterilize items within plastic bags, such as catheters, disposable items in labs and clinical settings, for example, packaged Petri dishes. And ethylene oxide is a form of cold sterilization, making it useful for the sterilization of heat-sensitive items. However, great care needs to be taken with the use of ethylene oxide as it is carcinogenic and highly explosive. So the sterilization is carried out in an ethylene oxide sterilizer, which resembles an autoclave in appearance. So this controls the ethylene oxide concentration, temperature, and humidity. Because pure ethylene oxide is explosive, it's usually supplied in a 10 to 20% concentration mixed with CO2. And the ethylene oxide concentration, humidity, and temperature will influence the rate of sterilization. The one big drawback that it has is that gas cannot kill dried up microorganisms and require a relative humidity level of 40 to 90% in the sterilizing chamber. And also, because it's so toxic, proper aeration of the products is required after treatment to remove the residual ethylene oxide. However, with careful use, ethylene oxide is highly effective, and ethylene oxide sterilizers are commonly found in medical setups to sterilize packaged materials. Okay, so the next group is the aldehydes. Formaldehyde is the most important aldehyde. It can be used in a special setup for gas sterilization, but its main use is in disinfection. Formaldehyde is usually dissolved in water or alcohol before use. It can, however, irritate the mucous membranes and the skin, resulting in inflammation or allergic eczemas. Glutaraldehyde is another aldehyde. that is used for disinfectant purposes and can also be used as a sterilant. A 2% buffered solution of glutaraldehyde is an effective disinfectant. It is less irritating than formaldehyde and can be used to disinfect hospital and laboratory equipment. Both of these chemicals are highly reactive and inactivate nucleic acids and proteins, and they are both sporicidal, so they both kill spores. So next we'll talk about alcohols. Alcohols are among the most widely used disinfectants and antiseptics. They work by rapidly denaturing proteins and may also dissolve membrane lipids. Alcohols are bactericidal and fungicidal, but do not destroy spores. They are less effective against viruses, but some enveloped viruses are also destroyed. The types of alcohol used in disinfection are ethanol, propanol, and isopropanol, used in concentrations between 60 to 80%. So alcohols act rapidly and penetrate the skin well, but they may cause skin irritation because they dehydrate the skin at the site of application. One common clinical use of alcohols is swabbing the skin to remove microbes before needle injection. Alcohols are also the active ingredients in hand sanitizers, which have gained popularity over recent years, especially at the moment. So on to phenols. So phenol, or carbolic acid as it's called, was the first antiseptic and disinfectant to have widespread use. In the 1800s, scientists began experimenting with a variety of chemicals for disinfection. And in the 1860s, British surgeon Joseph Lister began applying phenol to surgical wounds. So bear in mind that Lister worked as a surgeon at a time when sepsis, gangrene, and complications caused by infections were really common. So bed linens were not washed, and often surgical instruments were used to examine the wounds of every patient on the ward round before being wiped and put away at the end of the day. So no disinfection at all. And this is something we can't imagine these days. But Lister was actually the first person to apply the principle of the antiseptic barrier to a set of protocols for the operating theatre, and these protocols were designed to prevent microbes from coming into contact with the open wound. He introduced weak carbolic acid hand washes for the surgical staff and carbolic acid baths for the surgical instruments. He also used solutions of carbolic acid spray to reduce the level of the microbes in the air around the patient. So as the number of surgery-related infections fell, the evidence that antisepsis worked became undeniable, and it was widely accepted by the surgeons around the world. So Lister also inspired the American chemist Joseph Lawrence to develop Listerine. So that was named after Lister, and this is an alcohol-based mixture that is still used as an oral antiseptic today, which I'm sure you've all heard of Listerine. Today, carbolic acid is no longer used as a surgical antiseptic, as it is a skin irritant. But today, phenol and phenol derivatives, called phenolics, are commonly used as disinfectants in labs and hospitals. Triclosan is a bisphenol compound that has seen widespread application in antibacterial products over the decades, for example in hand sanitizers. Also, the commercial disinfectant Lysol is a mixture of phenolics. Phenols and phenolics work by denaturing proteins and disrupting cell membranes. So one common feature is they have a weak performance against spores and viruses. They also have the disadvantage of a disagreeable skin odour and can cause skin irritation, as mentioned previously. So next are halogens. So chlorine and iodine are important antimicrobial agents for disinfection, and they show generalized microbicidal effects and kill spores. And by microbicidal effects, we mean they kill microbes. Chlorine is the usual disinfectant for drinking water and swimming pools, but is also used in the dairy and food industries. It can be applied as chlorine gas, sodium hypochlorite, which is bleach, or calcium hypochlorite. And all of these yield hypochlorous acid in the presence of water. The result of this is oxidation of cellular material, the denaturing of protein, and the destruction of vegetative bacteria and fungi. Death of almost all microorganisms usually occurs within 30 minutes. Two important eukaryotic pathogens that are not killed by chlorine are Cryptosporidium and Giardia. Both of these are transmitted via water. Chlorine is also an excellent disinfectant for personal use. It's effective, cheap, and easy to use. Of note is the fact that household bleach, so diluted to 10% in water with 10 minutes contact time, can be used to disinfect surfaces contaminated with human body fluids. Iodine has similar qualities to chlorine and can be used as a skin antiseptic. It kills by oxidizing cell components and iodinating proteins. At higher concentrations, it may even kill some endospores. Iodine is often applied as a 2% or more iodine in a water ethanol solution of potassium iodide, and this is called a tincture. And this is used to disinfect skin and wounds. The problem with iodine is, although it's an effective antiseptic, it can damage the skin. A stain remains with it, and also people can develop iodine allergies. Iodine can be complex with an organic carrier to form an iodophore. And iodophores are water-soluble, stable, and non-staining, and they release iodine slowly to minimize skin irritation. They are used in hospitals for cleaning pre-op skin and in hospitals and labs for disinfecting. However, it's important to note that whilst the iodophores are less irritant to skin than pure iodine, they are also less effective as germicides. So the last group of chemicals that we're going to discuss are the oxidants and surfactants. So the oxidants include ozone, hydrogen peroxide, potassium permanganate, and peracetic acid, and their relative chemical activity is based on splitting off of oxygen. And most of these are used as mild antiseptics to disinfect mucosa, skin, or wounds. Next, go on to the surfactants. So surfactants include in soaps and detergents. They lower the surface tension of water to create emulsions that mechanically carry away microbes. There are different types of detergents available: anionic, cationic, amphoteric, and non-ionic. So just to remind you of those definitions, if you can't remember from chemistry. So anionic detergents have a negatively charged anion at one end attached to a long hydrophobic chain, whereas cationic detergents have a positively charged cation instead. Amphoteric surfactants have a dual charge on their hydrophilic end, both positive and negative. In acidic solutions, the amphoteric surfactants become positively charged and will behave like cationic surfactants, whereas in alkaline solutions, they develop a negative charge similar to anionic surfactants. The most effective detergents are the cationic and amphoteric detergents. So cationic detergents include an important class of disinfectants and antiseptics called the quaternary ammonium salts. And these salts are similar to phospholipids with their hydrophilic and hydrophobic ends. They are therefore able to insert into the bacterial phospholipid bilayer and disrupt membrane integrity. Cationic detergents such as benzalkonium chloride kill most bacteria, but not M. tuberculosis or endospores. Their advantages include low toxicity levels, lack of odour, good skin tolerance, and a cleaning effect. Cationic detergents are often used as disinfectants for food utensils and small instruments, and as skin antiseptics. So in summary, microbial control is important to prevent contamination, spread of disease, and protection from pathogenic microbes. We've learned about various physical and chemical methods that are used to control microorganisms. We've looked at pasteurization, which is important for controlling microbes in milk. We've learned about autoclaving, so that moist heat sterilization, which is important for killing microbes in general. And we've learned about the various chemical methods used to control microbes. And based on the availability of microbes in the environment, we've learned that different methods can be used. So again, here's your reading list for some further reading, which will help you gain further understanding in the topics. So next up, we have a little bit of a recap activity. So question 1, what is the difference between the terms sterilisation and disinfection? Question 2, what are the 6 main conditions that affect antimicrobial effectiveness? Question 3, why are prions not included in the standard definitions of sterilisation, sterility? Question 4, what are the 3 main physical methods of sterilisation and disinfection? Question 5, which method requires longer exposure times, dry or moist heat? Question 6, how does dry heat kill microbes? Question 7, what is an autoclave? 8, how do UV rays kill microbes? 9, what are the 2 main types of ionizing radiation that can be used for sterilisation? Number 10, what type of membrane filter would be needed to remove viruses from a liquid? So number 11, what are the disadvantages of ethylene oxide as a sterilant? Number 12, name 7 groups of chemicals that can be used for disinfection and sterilisation. 13, name an advantage of using glutaraldehyde rather than formaldehyde as a disinfectant. Number 14, describe the antimicrobial spectrum of alcohols. Number 16, what is triclosan? Number 17, what is a tincture of iodine and what is it used for? 18, what does chlorine produce when added to water? 19, what is a cationic detergent? Question 20, name a commonly used oxidant that is used as an antiseptic.